Hello students, welcome to the EPG Pathshala. My name is Dr. Shiv Kumar Sood. I am Senior Scientist in the Division of Animal Biochemistry at National Dairy Research Institute NDRI in Haryana, Karnal, India. I am going to talk about the module Molecular Sequence Databases from the paper Biostatistics and Bioinformatics. Molecular sequence data are non-linear sequences of DNA, RNA, and proteins. Functional information about these sequences may be derived from the sequence data itself. In addition, the sequence data also have attached useful information about these molecules, which we call annotation, is added manually by the database curators. All the information in the sequence and related annotations are stored in specific formats which are specific to the particular database provider. These particular databases have also developed the systems for the access and retrieval of the database and the sequence data respectively. Students will understand major online sequence gateways to retrieve and browse sequence data from various databases with different formats, and students will learn the conversion of one sequence format to other for use in other bioinformatics software tools. The bioinformatics tools enable biochemists to derive useful information from the nucleotide or protein sequence data for biochemical experiments and analysis. Therefore, nucleotide or protein sequence data is an important source for understanding the biochemical function of the genes and encoded proteins and RNA structures. In this module, you will be encoding linear sequences of the nucleic acids and proteins using single letter codes to the residues in the nucleic acids and the proteins. Student will be creating single letter coded sequence files using Notepad, an accessory available from Microsoft Windows operating system. This accessory is a simple text editor and can be used to create sequence files. So students will also learn different formats of sequence data for subsequent use by different software programs. You will have a look at various international public domain sequence databases, that is the archives, and the associated retrieval system used by these sequence databases as gateways for supplying the database sequence information. Students will also learn browsing the genomes for understanding the gene arrangement on chromosomes in individual organisms. Finally, you will be converting one sequence format into another using online sequence programs so that the sequence format recognized by a specific software can be obtained by just converting from one sequence format to another sequence format. The linear nucleotide or protein sequences are represented by single letter codes for the residues. The nucleotide or protein sequence data is stored as linear sequence of these single letter codes in the sequence databases. Recommended single letter codes for each of the 4 bases in DNA are like A is used for adenine, C for cytosine, G for guanine, and T for thymine. In RNA, U is used for encoding uracil. When we are not sure about the base in the sequence data, then the letter N or X is used arbitrarily. Similarly, there are recommended single letter codes for each of the 20 amino acids present in proteins. I'll give some of the tips for remembering single letter codes for all 20 amino acids. 6 amino acids, that is cysteine, histidine, isoleucine, methionine, serine and valine, are containing their first letter, which is not the first letter in other amino acids. Therefore, these first letters are used to store their names for encoding. However, when more than one amino acids begin with the same letter, then the letter is assigned to the most common amino acid. 5 amino acids, that is alanine, glycine, leucine, proline and threonine, are assigned their first letter because they are most commonly occurring amino acids. A for alanine is assigned because and not assigned to asparagine because alanine is occurring more commonly than asparagine. Similarly, glycine is occurring more commonly, therefore G is assigned to glycine, not to glutamine. Some single letter codes are phonetically suggestive. For example, F for phenyl, R for arginine, Y for tyrosine, that's why phonetically suggestive Y, and Q for glutamine. For 3 amino acids, that is aspartic acid, glutamic acid and lysine, a letter close to the initial is used for their code. For example, D is near to A, therefore it is used. E is near to G, therefore it is used for glutamic acid. K is near lysine, therefore it is used for encoding lysine. There are 2 peculiar cases in the amino acids. Asparagine contains N, and the N is not assigned to any other amino acid, therefore it is assigned to asparagine. Tryptophan is an amino acid which contains a double ring in its side chain, therefore W is assigned to tryptophan. B is assigned to either D or N, G means Z is assigned to either E or Q, and X for any amino acid. Specific bioinformatics software packages and online tools can open the sequence data files in their recognized formats. This is similar to opening an MS Word file with MS Word only. This file in MS Word cannot be opened with Adobe Acrobat Reader or say any other software because the MS Word format is not supported by those applications such as Adobe Reader. Similarly, reverse is also not possible. That is, MS Word cannot open PDB file saved by Adobe Reader. Therefore, a given software will open files with supported and recognized standard formats. However, this is to make clear that plain text saved in MS Word file is not a sequence format. It's supported by various bioinformatics tools. There are several specific sequence formats which are available, which can be used to save and store sequences. To save sequence in files, we need to provide 2 values in Save As dialog box. The first is the name to specify the primary name of the file, and the second is Save As type to specify the extension name of the file. Both names are joined automatically using the dot, that is a full stop or a period, for saving the file as by clicking Save As Save button. To understand sequence formats, let us take an example using Notepad, an accessory available with Microsoft Windows operating system. Enter the sequence of a protein using single letter codes in plain text and then select Save As command from the file menu. Type the name of the file as My Sequence. but use the default save as type text document, that is star.txt, in the save as dialog box. Then the sequence will be saved as file mysequence.txt on the hard disk. When we use mysequence.txt file name having .txt as extension name, it is not recognized by sequence analysis programs. Even if the mysequence.txt file is opened with a sequence analysis program, even then the plain sequence format is not recognized, cannot be read. Therefore, plain sequence in this file cannot be read with or used with any sequence analysis software. However, some online sequence analysis programs allow pasting of the plain text sequence in their input text box. To understand the meaning of sequence formats, let us see the most commonly used standard sequence format known as FASTA format. The sequence in the FASTA format can be saved even with Notepad or any other text editor. But there are two steps. The first is to enter the actual data, that is the annotation information and the actual sequence data in single letter codes. Second is procedure step to save this in a file as FASTA file using save as command of the file menu. The sequence information is typed in two lines. The first line is known as comment line, starting with greater than size. The greater than sign in the first line of a file shows that it is FASTA format file. After typing the greater than sign, type annotation information and comments in the same line. Annotation is usually some identification name and functional information for the sequence. Suppose we have a sequence with the name mySequence. For identification of this sequence, in the notepad, type mySequence after typing greater than sign. Continuing in this comment line, enter any other information such as annotation features, that this protein was sequenced using mass spectroscopy. Continue in the first annotation comment line, but without without pressing enter key on the keyboard. This shows that entering information will continue in the same line. As we enter the information in the comment line, we see that the initial information which we entered at the beginning of the line is not visible. To view the whole line in one window, select word wrap command from the format menu. This will display the complete entered information as one paragraph, but displayed in multiple rows, 3 rows in the present case. So this comment line is actually one single paragraph which may occupy multiple rows on the computer screen as seen, but actually it is single line. This comment line contains 3 pieces of information which are separated by a delimiter character, backslash. 3 pieces of information include the name of the sequence, then the source from which this protein sequence was isolated, and finally the technique used to sequence this protein. After entering this information, press the enter key so that the cursor moves on to the next line. In the next line, which is equivalent to next paragraph, the sequence of the protein or the nucleic acid is typed as shown for this protein sequence, which is this is the sequence of my protein. Then save the file by opening Save As dialog box of the file menu and enter file name mysequence.fa and select all files from Save As type and finally click Save button to save the file in FASTA format. Then to open the saved file mysequence.fa, select all files in the Open File dialog box as shown with arrow and click Open button. What is important in Open File dialog box is the drop down list all files. Always select all files from the choices in this drop down list, even when the FASTA format option is not listed in this drop down list. This will open the saved file. Now this file mysequence.fa can be used with any software package recognizing the FASTA format. This file can be used with any other text editor such as MS Word, which recognize the text stored as plain text in ASCII or ANSI format. Therefore, the file mysequence.fa can be opened even with MS Word after selecting all files in the Open File dialog box of the MS Word. The file will be displayed in the text window and user can select the sequence as highlighted in light blue in this slide. After selecting the sequence, click on the words in the bottom status bar to check the word count in this sequence. This will open word count dialog box from which it is revealed that the length of this sequence is 28, that is, there are 28 amino acids. In addition to entering single sequence in one FASTA file, one may add any number of sequences in one file in the FASTA format. Simply press Enter key after the first sequence to move on to next line. In the next line, type the comment starting with greater than sign and then again press Enter key to go to the next line and enter the single letter coded sequence without pressing Enter, as shown for the second sequence in this slide. In this way, one can concatenate as many sequences in one file in FASTA format as one wants to analyze. This is useful for pairwise and multiple sequence alignment as well as phylogenetic analysis. The International Nucleotide Sequence Archives Collaboration is the main database of nucleotide sequences, having 3 collaborators. First is GenBank at National Center for Biotechnology Information in United States of America, that is NCBI. The second is DNA Databank of Japan, and the third is European Molecular Biology Laboratory, that is EMBL at European Bioinformatics Institute. These 3 organizations exchange data on daily basis. NCBI integrates nucleotide sequence data with other databases for search in an integrated manner. Similarly, we have the Universal Protein Resource, that is UniProtKB, a comprehensive archive of protein sequences. In addition, Independent Protein Sequence Gateway at NCBI can be reached directly. XPASI, that is Expert Protein Analysis System, available at Swiss Institute of Bioinformatics, integrates UniProtKB databases with other protein information databases for searching in an integrated way. Each of the sequence gateway provides retrieval system separately for nucleotide and protein sequences. Entrez at NCBI, Expert Protein Analysis System XPASI at Swiss Institute of Bioinformatics, Sequence Retrieval System SRS at EMBL and dbGet at DNA Data Bank of Japan are main retrieval systems. In addition, we have integrated genome browsers for individual organisms, where user can have both the gene and the protein sequences with additional annotated information in an integrated way. The main genome browser is available at University of California at Santa Cruz. Visit NCBI and select Nucleotide in the left drop-down list of databases. To search, enter HPR from Enterococcus faecalis in the input text box to take an example and click search button. We find that there are a large number of records to be displayed. This is a long list to browse, but the first record is for Enterococcus faecalis of PTS-H gene encoding HPR. Click on any hyperlink in the list to display the data in GenBank format. However, if we are specifically interested in only searching for the data values, say HPR and Enterococcus faecalis, then advanced search shall be used. Therefore, click on Advanced Search feature available below the search text box. In the Builder section of the ensuing page, select fields to search and enter data values to be matched. First, select title as field and enter HPR as value. Then select organism as field and enter Enterococcus faecalis as value. Now click on the search button. The ensuing results page displays only one record in GenBank format. The GenBank format has three sections. First section, as shown here, is the header section with general information about the locus, source organism, literature, reference, and other information. The second section in GenBank format record is feature section, which contain gene and coding sequence, that is CDS information, having links to external databases. In this slide, an external link P07515 for UniProtKB SwissProt protein database is highlighted in yellow. Therefore, we'll use this in subsequent slide and reach in the UniProtKB SwissProt protein sequence database. Slide 27, and finally in the GenBank format, the section is sequence section. It starts with origin field and the sequence is displayed. The sequence is displayed in 6 columns, each column having 10 bases, that is 10 residues, which are numbered on the left side, starting with 1. To download the sequence, click on Send button and then choose destination and then the desired format, say FASTA in the present example. Click on Create File button and save the file in FASTA format using HPR efacalis.fa as extension name and selecting all files in Save as Type drop-down list. Even if the selected format for the sequence was any other, say for example GenBank, then one would enter the full name as hprefacalis.gbk as it was entered for FASTA format hprefacalis.fa. And now similarly select all files in the Save as Type drop-down list before clicking Save in this dialog box. Before 2002, Protein Information Resource, that is PIR, produced the protein sequence database named PIR PSD. SIB, that is Swiss Institute of Bioinformatics, produced manually curated SwissProt, and EMBL produced computationally translated coding sequences database, that is Tremble, translated EMBL, awaiting manual annotation for inclusion into the SwissProt. In 2002, these three institutes pooled their resources and produced what we call today UniProt knowledge base, that is UniProtKB. It has two components, UniProtKB SwissProt, that is the manually annotated component of the UniProt. The manually annotated component contains reviewed and annotated proteins with information extracted from the literature and the curator evaluated computational analyses. On the other hand, UniProtKB Tremble is computationally analyzed proteins, that is, which are yet not reviewed and will be reviewed later on, and then after review they will be transferred to the SwissProt component by adding annotations. So UniProt, SwissProt and UniProtKB Tremble are two components in the UniProt consortium. Let us return back to GenBank sequence format for using external links. One can reach on any of the external links listed on this page. In the present example, the external link to UniProtKB is highlighted in yellow. This is the link for protein in the SwissProt database. Just click on this P07515 external link to reach UniProtKB sequence record. The most important in UniProt record is the display menu on the left, in the left column. One could jump to any of the feature just by clicking in these hyperlinks. The features include function, names and taxonomy, subcellular location, post-translational modifications and processing, interactions with other proteins, 3D structures, conserved families and domains, sequence and external links to other sequence databases, publications and literature information. One can visit UniProtKB directly at uniprot.xpsg.org and search for HPR from Enterococcus faecalis. In the results page, click on Reviewed as shown by the arrow in this slide. This will display only reviewed SwissProt sequences. To download a sequence in FASTA, open the Download tab as shown and select FASTA canonical format and click Go. The FASTA sequence will be displayed in the browser window. This sequence may be selected, copied and pasted in Notepad window and then saved using Save As command of the file menu. Now let us use a genome browser. When the user is specifically interested in a particular organism, say Enterococcus faecalis, for which the complete genome sequence is available, the user shall get the nucleic acid and protein sequences as well as associated information using a genome browser. Visit Microbe's genome browser at University of California, Santa Cruz and select the group for bacteria, firmicutes and select genome organism Enterococcus faecalis. In the search box, input phosphocarrier protein HPR and press Enter key. You will reach the gene EF0709 encoding protein phosphocarrier protein HPR displayed in the genome browser window. Bring your mouse over the gene bar, which is cyan colored. This will display the gene name EF0709 and the product phosphocarrier protein HPR. To download the sequences, click on this gene bar. User will reach a page where links to sequence is displayed for predicted protein, predicted mRNA, and genomic sequence. Click on predicted protein. Your browser will show the protein sequence in FASTA format starting with greater than sign, then EF0709, length is equal to 28, followed by the sequence in single letter codes. Copy the complete FASTA sequence and save it as efcalis.hpr.fa using Notepad on your hard disk. Sequence formats were designed by specific database developers or groups or companies to hold the sequence data and related information to the sequences for use in their own programs and software packages. There are several sequence analysis software packages and tools which are available online for sequence analysis. A specific software package or tool will support only some recognized standard formats. This shows that there are several sequence formats. However, some sequence formats are internationally recognized as standard formats which are used most commonly. Almost every database of sequence such as GenBank, European Molecular Biology Laboratory EMBL, SwissProt at Swiss Institute of Bioinformatics, PIRSD at Protein Information Resource, etc., has stored their data in their own formats. These databases allow to download the sequence data in additional formats also. But in case we do not get sequence data in the desired format, then we have the option of downloading the sequence data in the format recognized and provided by the database developer. After downloading the data, sequence data in the format of the data provider, user can convert this sequence format into any other sequence format for use in other sequence analysis programs. In this last section of the module, you will learn sequence format conversion. To convert a sequence format, go to Sequence Format Converters at EBI and choose to launch Emboss Secret. Follow the 3 steps on the appearing browser window. First step is to upload already saved file GenBank HPR protein sequence.gbk in GenBank format and choose it to convert SwissProt entry format, Swiss new, and click Submit button. Another implementation of Secret Emboss is available at genome.nci.nih.gov/tools/reformat.html. Paste the FASTA sequence in the text box and then select the input sequence and the output sequence format from the drop-down list and click Submit Request button. The result will appear in the browser window, and the resulting window will display sequence of histidine-containing phosphocarrier protein HPR from Enterococcus faecalis sequence in SwissProt format. Dear students, in this module, you learned that there are several sequence analysis software packages and there are several online sequence analysis tools which require a specific sequence format for the analysis. You learned encoding linear sequences of nucleic acids, that is DNA and RNA, as well as proteins using single letter codes and saving them in FASTA format, most commonly used format, which can be created even using Notepad. Then you downloaded nucleic acid and protein sequence from GenBank and UniProt knowledge-based databases. You learned that in case user is interested in a particular species, say for example Enterococcus faecalis or E. coli or Homo sapiens, then in those cases it is best to use genome browser to browse genes on the chromosomes and download predicted protein, predicted mRNA and DNA sequence data directly using the genome browser. Finally, you learn to convert various sequence formats so that the sequence data can be used by the desired software analysis program. Thank you for visiting EPG Pathshala.